Morning, everyone. As Rick has said, this is a joint presentation between the Liverpool National Alkaptonuria Centre and the Liverpool School of Tropical Medicine. It's going to be a very quick overview because each of us, I guess, could spend talking about our topics for half an hour each. So you're just going to get the broad brush of what we're about. What I would like to start with is to say that this is a really repurposing issue when we started. There was nothing in the UK for Alkaptonuria, a rare disease, ultra rare disease, with a frequency of 1 in 250,000 population. When we started in 2003, there was nothing. And we founded the AQ Society in 2003, and you can see the founders on the left side there, and the CEO. We got going, and this is how the whole thing started. Alkaptonuria is a deficiency of the enzyme homogeneous dioxinase. What that means is this is a tyrosine pathway responsible for breaking down most of the surplus phenylalanine and tyrosine that we consume. So the lack of this enzyme means you can't break down homogeneous acid, which accumulates and builds up in the body, so much so that it's excreted very efficiently in the urine, thankfully. It colors the urine black from birth, so it's easily spotted. And despite that, there's accumulation of homogeneous acid in the circulation. Tissues are prone to have HGA in them, there's deposition, and it alters the nature of the tissue so that they become stiff and brittle and break down and cause the multi-system disease, which is Alkaptonuria. Our goal when we started was to try and use a drug that has already been in use for a condition called hypertyrosinemia type 1 since 1991. And this drug was called nitisinone. This drug inhibits the conversion of para-hydroxyphenyl pyruvate to homogeneous acid. And it's directly proximal to where we want it to act. And our hope, which in fact was realized, is to say that by lowering the homogeneous acid, we could decrease the ochronosis, and therefore we could decrease the disease. This is a timeline of some of the things we did between 2003 until we really got going in 2012. I would love to tell you about all the things the pioneers were working on it before 2003 and also until 2012, but time doesn't permit. So I'll go straight to 2012. Two important things happened. One was the creation of a national centre for Alkaptonuria in Liverpool by NHS England, highly specialised services. And the second thing was the European Commission funding for a series of clinical studies in Alkaptonuria called Develop a Cure. The culmination of Develop a Cure was a 4-year clinical trial called SONIA 2, Suitability of nitisinone in Alkaptonuria 2. Suffice it to say, both of these, this is the National Centre data, showing that off-label use of nitisinone was successful, as well as the Develop a Cure 4-year clinical trial showed that nitisinone was effective and safe in Alkaptonuria. The culmination of that, of course, was the European Medicines Agency approval of nitisinone as the first treatment for Alkaptonuria in 2020. This is actually seems like a long road since 2003 to 2020, but it's actually quite quick if you really think about it. But that's because of the fantastic collaboration we had between the clinical scientists, academic scientists, and the patient societies. Rare is fundamental. Why study rare diseases? So I'll give you 2 examples. One of these refers to this patient of mine, who is a 50-year-old man who had lancinating hip pain, and he had joint surgery as a result of that. And when the hip joint was taken out, you can see there that the hip looked pigmented but relatively normal. There was no loss of cartilage. So we didn't understand why on earth he was having this pain. So really careful studies showed that he had projections of highly mineralized, if you like, needles sticking out from the articular cartilage, the subchondral bone, as you can see there. So this is spongy hyaline cartilage, and this is the calcified cartilage. And you can probably see the crack there through which bone marrow fluid leaked out into the spongy cartilage and then became highly calcified. And it's like having a needle stuck in your joint. And this was causing the pain for him. And after the surgery, he was fine and mobile and active. So based on this, we came up with the idea of, you can see it better now, it's enlarged. Based on this, we have a hypothesis by which we think the brittle cartilage, the calcified cartilage in the subchondral bone develop cracks through which fluid leaks out and then forms these needles that cause the pain. We thought initially this was rare and occurred only in Alkaptonuria, not necessarily. So we've been looking for this now in osteoarthritis, and we have found these needle-like projections shown by the yellow arrow there on the MRI of osteoarthritis, saying that this phenomenon occurs even in osteoarthritis, which affects all of us. And of course, it also made the late press, Weber-Clast. And this has been led by my colleague from the University of Liverpool, Professor Jim Gallagher. I'm just going to give you two more important examples. So repurposing nitisinone is really fantastic, and I hope to convince you of that. The way nitisinone works, I've already told you, blocks the HBPD enzyme, and therefore decreases the metabolites distal to the block. It increases metabolites proximal to the block and flux through the pathway. So if you look at the phenylalanine tyrosine pathway, what I guess I'm saying is, so this is the block, these are decreased, and this increases, and there's increased flux in the dopamine and the melanin pathway. I'm going to tell you the starting point for this, which was nitisinone was developed as a herbicide. And of course, what you see there is the bleaching reaction that nitisinone produced, because in plants, the homogenic acid becomes plastoquinone, which is part of the chloroplast needed for chlorophyll synthesis. And of course, if you block that pathway, then of course you can't make chlorophyll and the plants die. So that was the way in which the mode of action of nitisinone was understood at the time. And of course, I've already told you that this was the pioneering work done by Scandinavian scientists and Dr. Edward Locke, Professor Edward Locke, who lives in Liverpool, by the way, who made nitisinone available for the treatment of hereditary tyrosinemia type 1. And this completely changed the treatment for this condition, because his liver transplantation was the treatment for them at the time. So it is life-saving treatment that these scientists managed. However, I already told you about the repurposing of nitisinone in Alkaptonuria. It's been suggested it could also be used for Hawkinsonuria. And of course, going on to the flux, you can see this is a nitisinone has been used to prolong life in advanced neuroblastoma. It has also been tried, as you can see there, in restless leg syndrome, which is highly common, supposed to be sleep disorder, occurring in 5% of the population. It's also been trialed in the National Institute of Health in the US in oculocutaneous albinism B with some success. And it has also been suggested as a treatment in Parkinson's disease, because it's a bit like giving dopa to patients, except that it's central. And finally, we all know about phenylketonuria. Phenylketonuria is much more common than alkaptonuria. And the problem in phenylketonuria is that you can't convert phenylalanine to tyrosine. So many people believe that the features of phenylketonuria is to deficiency of tyrosine. So therefore, this group have shown that nitisinone, by blocking the degradation of tyrosine, can also improve PKU in a mouse model. So the opportunities for repurposing are simply enormous. And I would love to call this multi-purposing rather than repurposing. rare is fundamental. So here's another opportunity for us as a group to understand a bit more about a more common disease. We found that in the National Alkaptonuria Center, the prevalence of Parkinson's disease is 20 times more common. Okay, and we don't know why. So, so this is the same pathway. So we think the reason is this compound called 6-hydroxydopamine. 6-hydroxydopamine comes from dopamine pathway. And we think in homogentic acid causes the conversion of dopamine, 6-hydroxydopamine, when Parkinson's disease occurs in AKU patients before they go on nitisinone. And in patients who go on nitisinone, it's the flux into the dopamine pathway also increases 6-hydroxydopamine. And of course, this has been touted as the reason why DOPA stops working in Parkinson's disease after a few years. And the reason for that is because you're also producing this neurotoxic 6-hydroxydopamine, which causes the DOPA to stop working. So we would love to look at the Parkinson's disease through the lens of AKU. And we're just about thinking about how to do that now. This is my last slide before I hand over. So nitisinone HT1 is really an amazing achievement by the scientists that are listed there, two from the Karolinska and Edward Locke, who also happens to be part of our group. And but Ted told me 2 or 3 years ago about some amazing work that's going on in the Liverpool School of Tropical Medicine, where they were using nitisinone. And at this point, I would like to invite Dr. Álvaro, Acosta, Serrano to come and talk about the amazing work that they have been doing. So there's the laser. That's
okay. Thank you so much, and thanks for having us here today. So we study insects and parasites that are transmitted by those insects. So that what does it have to do with nitisinone? And it turns out that this very talented man here, postdoc, he discovered that when insects, in order to transmit diseases, they need to take blood first, right? And that blood needs to be digested. That blood is essential for producing eggs, for example. But when he blocked that pathway, he realized that that was essential for those bugs. And when he blocked that pathway pharmacologically or using genetic tools, then he realized that was lethal to all these insects. Then later on, he joined forces with my colleague Haines, who will speak later. And we tested these in another bug, important one, tsetse flies causes trypanosomiasis in Africa. So the way it works is exactly as Professor Ranga described. So nitisinone, the pathway is present in all insects. And if we look into just a general metabolic analysis, so when nitisinone is present, it accumulates tyrosine. And then, for example, the end products like fumaric acid, it goes down over time. So you see the accumulation of tyrosine here, got the drops of fumaric acid. So how does that work? It's very different to any other insecticide compounds. Most of the insecticides used to kill bugs, they are neurotoxic. They kill an insect right away. In this particular case, it takes some time. And you can see a difference between tsetse flies that have been fed just with normal blood or blood with some nitisinone. And after a few hours, you can see that the tsetse flies start to paralyze, and then they just basically stop digestion and start dying basically right after this. And they present all sort of phenotypic changes, a change in eye color, for example. There is a darkness of the abdomen. So there's a process called melanization. Melanization is just the darkness of production of melanin internally in all tissues and outside, and that's what the colors become like this. If you dissect this insect, there is nothing left inside. So the beauty of this, and that's why we're pushing, is because it's a specific for insects that need blood to survive, okay? So pollinators, for example, like bees, they're completely harmless to nitisinone or any other drug that prevents or blocks tyrosine degradation. So in this particular experiment, we supplemented the sugar solution that bees leaves out with a really high concentration of nitisinone that we know kill mosquitoes or any other bugs quickly. And you can see compared to the control, they survive to a few days. So with that in hand, and given that at the Liverpool School of Tropical Medicine, we have a lot of experience in mosquito biology, mainly to prevent malaria, to develop new tools to prevent malaria in the field, we decided to test it for mosquitoes. And I don't have to probably remind you that mosquitoes are the deadliest animal on the planet. So it kills at least 500,000 people every single year of malaria, mainly children. So what we did is to use our experience with city flies, so we can use artificial membrane feeding systems like this. So we can feed the females with blood spike with nitisinone, for example. In order to do that, what we needed to do is to compare. We calculated based on the known PK and PD profiles of nitisinone, how much the concentration will be in blood over a period of a month. So the ideal drug to use in the field is a single dose that should last for at least 2 weeks up to 4 weeks. We don't want patients, we don't want people in the field to take drugs too often. Also, it lowers the cost. And logistically speaking, in areas that are deprived in poor resource settings, that's the best thing. So ivermectin, in comparison, is being used in the field. Ivermectin also kills mosquitoes. And we wanted to compare with the gold standard. And you can see that the PK profile and the PD profile is actually quite better, much better compared to ivermectin over time. So then we use those calculated concentrations to feed mosquitoes in the laboratory. And you can see here is that the mosquitocidal effect with nitisinone compared to ivermectin is much more superior. Okay, so we then joined forces with Professor Ranga. We were doing all this work without knowing his existence, what we were doing with our Alkaptonuria trials in Liverpool. So that was actually a great coincidence. And among the many things we'll be doing, and that's part of Anna's PhD work, is to take blood from the Alkaptonuria patients that are currently being on these nitisinone trials. And just to give you an example, for example, this is the 4 different patients in here, okay? In blue, you see the mosquitocidal, the killing effect with neat blood or just 50% diluted. You see that there is no differences. And then when we dilute a little bit, obviously, it lowers the effect. In here, you can see an Alkaptonuria patient who has not received nitisinone, meaning that the killing effect is completely due to nitisinone, not to any metabolite produced during Alkaptonuria, and those are positively controlled for our assay. So that's a proof of principle that when humans take the drug remains active, and it's actually even more active than we thought it'd be. So there are many ways to use nitisinone. So I'll show you an example of the oral drugs just to make a link with Professor Ranga's talk. But we know that it also can be solubilized in organic solvents. So it could be used as insecticide because it goes through the cuticles of the insect. It could be used as what we call sugar bait. This is just a sugar solution. So mosquitoes, for example, they get attracted to it. It's important for the biology. They take a sip of that sugar solution with nitisinone, and the moment they take a blood meal, they die. So, and another aspect of this is the fact that most of these vector-borne diseases are zoonoses. So they infect animals, and including farm animals. This is one of the main reasons development in Africa is affected. But if we treat animals that will also benefit humans in different ways, we can then minimize the effect, the impact of it. And that is important because a lot of our countries in sub-Saharan Africa has these overlapping regions with malaria and other vector-borne diseases. So again, we know that we can use this to complement current strategies for preventing malaria in the field. Malaria continues to be the deadliest disease on the planet. If we take about beside pandemic time, But then there is another interesting aspect. Many more vector-borne diseases that are not lethal, they're really conditions that affect humans in many ways, okay? And we're talking about mainly poor countries, probably a quarter or more of the population, per couple of billion people, are affected by what we call neglected tropical diseases. And they have really horrible conditions that invalidate the life and prevent society in many ways. So with that in mind, I'm just going to pass on to my colleague Lee Haines, which then will emphasize why all the work done in rare diseases could have a huge impact for preventing malaria and other vector-borne diseases.
Good morning. My name is Lee Haines, and I imagine at this conference you never thought you'd be learning about insects. So that's your surprise today. This is just to give you an overview of what Professor Ranga and Dr. Acosta Serrano was saying, because I think having two talks side by side sometimes needs a summary of what we were doing. And I was working on this project for almost 6 years now at the Liverpool School of Tropical Medicine. So I just want to say that nitisinone is a fantastic drug that we're working with from a global perspective. And it all started with Ted Locke working with plants. And it's gone to insects and humans. So it's really quite a large variety of life forms that it affects. So it starts out as a plant, as a herbicide. It causes photobleaching, the plants die. We then thought, okay, let's see if this can be repurposed through the delivery of it in blood to insects, because we knew the pathway was important. And we also, because of Ted Locke, were introduced to Professor Ranga and moved into the rare neglected disease environment. Professor Ranga has shown you how it's worked with tyrosinemia type 1, Alkaptonuria, and the other diseases that he's flagged. So both PKU, neuroblastoma, all of these exciting ones where there's so much more research to be done. But I wanted to just highlight what we are working on, which is looking at how to affect the global distribution of this disease, of this drug. And here, if you were focusing on rare neglected diseases, we're looking at a single person or a very small population of people, which means the drug cost is really high. And if we were to expand this and look at, for instance, the neglected tropical diseases, this opens a whole new idea for users and therefore dropping the disease price or the drug price. So Álvaro's already mentioned malaria, which is a very large disease across the world. It's not a neglected tropical disease, but what we do have is lots of insects, and we call them vectors, that spread these diseases. So we have tsetse flies, sand flies, kissing bugs, ticks. Lyme disease is one that is here in the UK. Arboviruses are things like viruses that are Zika or chikungunya or dengue virus, ones that are spread by particularly mosquitoes. And when you consider all of these different disease vectors and the use of nitisinone in all these different ways that Álvaro's mentioned, whether by the mosquitoes touching or ingesting the blood, you end up with a lot more people that could possibly be protected. And when you do this, you realize that once you have a large group of people to be helped by a drug, you will drive down the price overall, which means it's more affordable for people in other countries to use. And also it has a much larger remit, so global distribution. So that's all I wanted to say about this summary. It's an amazing drug that looks like it can affect many different people in a positive way. And just to highlight that we are not here on our own. This is a huge team effort from the Liverpool School of Tropical Medicine, the Liverpool University Hospitals, all of our funders. And really, I want to highlight Ted Locke at Liverpool John Moores University, who brought us all together through collaborative discussions, which are unusual. So I think when we talk about rare diseases and how we make progress, is going beyond our boundaries. I'm a vector biologist, Álvaro's a parasitologist, Ranga is everything. So really engaging with discussions and pulling this out is how we're going to make really strong strides forward. So thank you.